Hello everyone, today we are going to discuss about the industrial production of streptomycin. It is an antibiotic as we all know that. And next introduction for streptomycin. Streptomycin is the first antibiotic discovered and it is produced from the bacterium Streptomyces gracius. The bacteria is found in the soil and Streptomyces species is gram-positive bacteria and it grows in various environmental conditions. It is filamentous in nature. Streptomycin was discovered by Albert Schatz, Bugey, Waxman from Streptomyces gracius in the year 1943. It is primarily used for the treatment of tuberculosis and for the bacterial infections caused by gram-negative bacteria. It is known to be a broad-spectrum antibiotic. So it involves the production of streptomycin involves various stages from the microbial production, fermentation to purification. And next, microorganism selection. The first step in the industrial production of streptomycin involves the selection and cultivation of suitable strain for the production of streptomycin. For this, Streptomyces gracius with efficient strain is selected. And next, inoculum preparation. The selected strain is grown in a small scale seed culture in the liquid media to prepare an inoculum for large fermentation tanks. The inoculum usually consists of Streptomyces spores or a small amount of liquid culture containing active bacteria. Next, the preparation of medium, a media which supports the growth of microorganisms which contain essential nutrients and other minerals and growth factors to support and enrich the growth of microorganisms we are going to produce. So first, carbon source is used as dextrin, glycerol, glucose, starch and other substances. And nitrogen source, naturally occurring processed agricultural products such as cottonseed meal, soybean meal, constip liquor solids, casein and hydrolysates. Yeast and its prepared extracts also act as a nitrogen source. And pure inorganic salts such as ammonium sulfate, ammonium nitrate is used. And vegetable or animal fat, soybean oil, linseed oil, fatty acids are used. Fermentation condition during the production of streptomycin. The temperature should be maintained between 26 degrees Celsius to 30 degrees Celsius to carry out the production of streptomycin. And then the pH should be maintained between 6.8 to 7.2. So it is optimal growth condition to support the production of streptomyces, which supports the antibiotic biosynthesis. And next, aeration and agitation. So aeration is necessary because Streptomyces gracius is an aerobic organism and oxygen is required for the bacteria's metabolism, and the fermenter is equipped with the stirrer which mixes the essential nutrients present in the medium. And the fermentation duration. Usually the production of streptomycin takes about 4 to 14 days. It depends on the strains we use and media composition and other conditions. During this period, the bacteria grow and produce streptomycin which accumulates in the fermentation broth. And antibiotic production phase. So initially the bacteria use the nutrients in the medium for their growth, but once the growth phase slows down, the antibiotic production phase begins. At this stage, the bacteria focus on producing secondary metabolites like streptomycin, and regular monitoring of the concentration of streptomycin in the broth should be essential to determine the optimal harvest time. And next, harvesting the fermentation broth. After the fermentation process is completed, the broth need to be harvested. The broth contains a mixture of streptomycin, bacterial biomass, metabolites and residual nutrients. For separation of biomass, the first step is to separate the microbial biomass from the liquid fermentation broth. For this, centrifugation or filtration techniques can be used. The bacterial cells are then removed. The cells are harvested and the streptomycin is need to be purified by various methods. Solvent extraction. Streptomycin can be extracted using various organic solvents and then precipitation. So the streptomycin can be precipitated from the broth by adjusting the pH or adding specific salts. And then chromatography. Chromatography techniques like ion exchange chromatography or gel filtration chromatography can be used. For this, the particles are separated based on the ionic interaction. And then crystallization. After purification by chromatography, the solvent extraction, streptomycin can be crystallized from the appropriate solvent. Crystallization is a final step in obtaining pure form of streptomycin. And the final step is formulation. After purification, the antibiotic is usually in the form of crystalline powder and it can be formulated as injectable solutions, which is used for treating severe infections like tuberculosis, and then into a oral form. It is formulated into tablets capsule for oral administration. During formulation, excipients are added to stabilize the antibiotic and ensure its bioavailability. These can include stabilizers, buffers, and preservatives. Thank you all.